Hi, my name's Clement Savage. I do biology at A level, and today's video is going to be on a little bit on chemosmosis, but mostly on oxidative phosphorylation. And we should be able to, by the end of this video, outline the process of oxidative phosphorylation, be able to reference the role of electron carriers, oxygen and mitochondrial cristae, state oxygen as the final electron acceptor. Before you watch this video though, go back and watch my video on chemiosmosis. They're basically the same video, just cut into two parts, because my stupid uploading thing isn't working that well. So where we finished off in chemiosmosis is the hydrogen ions were not able to diffuse through the lipid part of the inner membrane, but they could diffuse through the ion channels in it. And those channels were associated with ATP synthase, and that flow of hydrogen ions is chemiosmosis. So now we're going on to oxidative phosphorylation, and this is the formation of ATP by the addition of inorganic phosphate to ADP, okay, not TP, ADP, in the presence of oxygen. And this happens because protons flow through an ATP synthase enzyme, and this drives the rotation of the part of the enzyme. And this joins ADP and PI, which is inorganic phosphate, I know it's the wrong way around, and PI to form ATP. And these electrons are passed from the last electron carrier in the chain to molecular oxygen, which is the final electron acceptor. Hydrogen ions also join so that oxygen is reduced to water. So, before oxidative phosphorylation, how much ATP was made? So far, for each glucose molecule, 2 molecules of ATP have been gained during glycolysis by substrate-level phosphorylation. And during the Krebs cycle, 2 molecules of ATP were made by substrate-level phosphorylation. So that's how much ATP was made before, but how much was made during oxidative phosphorylation? Well, more ATP will be made during oxidative phosphorylation, where the reduced NAD and FAD molecules are reoxidized. The reduced NAD and reduced FAD will both provide electrons to the electron transport chain to be used in oxidative phosphorylation. And reduced NAD also provides the hydrogen ions that contribute to the build-up of the proton gradient for chemiosmosis. The hydrogen ions from reduced FAD stay in the matrix, but can combine with oxygen to form water. So, more ATP made during oxidative phosphorylation, where reduced NAD and FAD molecules are reoxidized. These produce electrons to the electron transport chain. Reduced NAD also provides hydrogen ions. These contribute to the build-up of the proton gradient for chemiosmosis. Hydrogens from reduced FAD stay in the matrix, but can combine with oxygen to form water. And my sisters, sorry. The 10 molecules of reduced NAD can theoretically produce 26 molecules of ATP during oxidative phosphorylation. And therefore, for each molecule of reduced NAD that's reoxidized, up to 2.6 molecules of ATP should be made. So together with the ATP made during glycolysis in the Krebs cycle, the total yield of ATP molecules per molecule of glucose should be about 30. It should be 30 really, yeah. But this isn't always, well, this is rarely achieved. And this is a question that could come up in the exam, like why isn't the maximum yield always attainable? And that's because some protons leak across the mitochondrial membrane, because could have a leaky membrane, and this reduces the number of protons to generate the proton motive force. Some of the ATP produced is used to actively transport pyruvate into the mitochondrium, and some ATP is used for the shuttle to bring hydrogen from reduced NAD made during glycolysis into the cytoplasm into the mitochondria. So if you just learn one of those, really, you'll be able to answer the question. Also, always refer to protons being pumped into the intermembrane space. Don't say they're actively transported, because this implies that they are using ATP, and in this case the energy is from the electron flow, not from ATP. So that is basically finishing off chemiosmosis and oxidative phosphorylation. Some questions could be: explain why oxygen is known as the final electron acceptor. Explain why the proton gradient across the inner membrane is a source of potential energy. Any of those. Well, this is quite a short video. Watch it together, the other one, and you'll be fine. And good luck in your biology exams.